My name is Elaine Bignell, and yes, I'm a former member of the Manchester Fungal Infection Group, but I moved to the University of Exeter during the first lockdown in 2020, and I'm now a research co-director at the MRC Centre for Medical Mycology, which is based on this beautiful Streatham campus at the University of Exeter. And our centre is based in the Geoffrey Pope building, which is sort of perched on top of the hill here on the campus. Now, I'm a fungal geneticist by training. What that means is that I'm an expert in fungal genetics, and I'm an infection biologist by research. My interest is in fungi that thrive in our natural environment, but sometimes cause very serious human diseases. And in this slide, I wanted to highlight, just by showing you these two very different images of the campus, the very important role that environmental fungi play in the annual turnover of organic matter. And of course, the truth is that fungi are all around us. And every breath that we take is full of fungal spores. In fact, with the obvious exception of the commensal yeasts that live on our skin and in our guts, the overwhelming majority of fungal organisms that can harm a human being are acquired via the inhalational route. Now, if we think about global burdens of severe respiratory infection, there are a number of important factors that we should consider. One of these is how frequently these diseases occur. Another is how many of those infected will suffer serious disease or even die. And the number of serious diseases occurring is the product of these two factors. And we often use these metrics to measure how well we're doing with tackling particular diseases. So, for example, in 2020, there were over 100 million SARS-CoV-2 infections reported, and these led to 1.8 million deaths, a case fatality rate of 1.8%. This is a better management scenario than that of TB that caused a similar number of deaths that year but affected far fewer people. And it's worse than malaria, where many more people were affected but far fewer died. The problem of invasive fungal infection is just as serious as these well-known diseases, but a lack of investment into this area has kept fatalities high, particularly in the low-income countries where access to existing drugs is a challenge. And we're now also facing big challenges with the resistance of some fungal species to the handful of available drugs. And you've heard about our colleagues at the Manchester Fungal Infection Group there highlighting that problem. So the Centre for Medical Mycology was established in 2019 to tackle these challenges by building capacity in basic and clinical mycology expertise. And research in the centre now covers 5 research themes that collectively seek a better understanding of fungal diseases and seek to drive the development of better diagnostics and antifungal therapies that can improve their prognosis. And the work of the centre covers a vast range of fungal pathogens. In the talk today, I want to introduce some of the CMM researchers who work on the problem of Aspergillosis. And this group includes scientists from every career stage and individuals whose work addresses research across the 5 themes of our centre. And throughout this talk, they will tell you in their own words how the work that they do seeks to improve our understanding and ultimately the outcome of these devastating diseases. So around the globe, the common mould Aspergillus fumigatus accounts for a very significant proportion of fungal respiratory disease. And this fungus is a soil-dwelling mould, and it sporulates abundantly, and it grows very rapidly at 37 degrees, which is human body temperature. So the fungus propagates by releasing these spores from a specialised vertically growing fungal cell, which is called an aerial hypha. And these spores are dispatched from the aerial hypha, and they're designed to withstand stress and to fend off natural predators in the environment until they find a new moist and carbon-rich environment. And at that point, each of those spores will undergo a new program of growth, involving the swelling of the spore, germination, and then rapidly branching growth of these elongating cells called hyphae. And that's what we're watching in this video in the bottom left corner here. And these extending hyphal tips release a cocktail of enzymes and toxins that drive damage to host tissues and blood vessels. And these growth behaviours that we can image in the lab are very similar to those that we observe in the tissues of mammalian hosts infected with Aspergillus fumigatus. So Aspergillus species cause a range of fungal lung diseases in various clinical settings, and these diseases vary. They can be rapidly fatal, chronic or allergic in nature, and we now know that we can include severe viral pneumonias in the list of risk factors for invasive aspergillosis. Now, to bolster our clinical expertise in this area, the Centre for Medical Mycology has been awarded funding to train a dozen or so clinical research fellows over the lifetime of its MRC funding. And these research fellows undertake PhD studies out of their clinical training programme, but alongside ongoing clinical study. And their research addresses aspergillosis in the clinical settings in which they work, and will return to work in after their training. And the aim is to achieve a better diagnosis and management of aspergillosis in these settings.
Hello, my name is Emily Cheshire. I am a paediatric consultant at the Royal Devon and Exeter Hospital, and I am a clinical research fellow at the Centre for Medical Mycology. I'm studying towards a doctorate in medicine with Professor Adelia Warris, in which I am investigating the role of Aspergillus infection in children and young people with cystic fibrosis. This is a really important area, and I'm hoping to understand more about how common Aspergillus infection is and its long-term consequences in this group of patients.
Next video, Graham. Yeah.
Hi, my name is Laura Ferreras. I'm a pediatrician, and I'm also working at the MRC Centre of Medical Mycology of the University of Exeter. Aspergillus can cause very severe infections in vulnerable children, mostly those who have cancer or those who have undergone a bone marrow transplant. My main research interest is to try to improve how we diagnose these infections in this group of children, and also how we use better the treatments we have. So only those children who really need them receive these drugs, and we avoid the development of resistances for the future.
Great, next slide. Thanks, Graham. Okay, so across the spectrum of invasive and chronic diseases that I spoke about at the beginning of this talk, frontline therapy relies upon a single class of drugs called the azoles. And as we've heard from multiple speakers in the past hour, the dual use of azoles as medicinal and agricultural fungicides has really accelerated the emergence of clinical resistance to these drugs. And what we're looking at on the left is work by Matt Fisher and colleagues at the MRC Centre for Global Infectious Disease Analysis, who are collaborating with scientists in Manchester and at our centre in Exeter to lead global efforts to track and trace the ecological and clinical drivers of this antifungal resistance problem. And we know that this is a problem that has a measurable human cost, as evidenced from data on the right from our colleagues in the Netherlands, who've been able to show that in patients who are infected with isolates that are voriconazole resistant, there are worse outcomes from treatment in those groups. Next slide.
One click.
One click.
There you
go. Okay. Right. So the critical problem of antifungal drug resistance is a standalone research theme at the Centre for Medical Mycology in Exeter, and much of our centre's research touches upon this topic because by understanding the fundamental biological processes that cause fungi to become resistant to existing antifungal drugs, we can better design and use medicines to circumnavigate that problem. So Darren Thompson in our centre has really championed the use of live cell imaging approaches to observe and document the biology of fungal cells as they interact with immune cells from the human host or when they are challenged by antifungal drugs. Next video, please, Graham.
Hello, I'm Dr. Darren Thompson at the MRC Centre for Medical Mycology here at the University of Exeter. And my role here is to develop and maintain advanced fluorescence imaging technologies, which allow us to observe the infection biology involved between host and fungal pathogens, such as the green Aspergillus fumigatus spores and germlings up here, and the cyan epithelial here. And although this is a snapshot of what's going on during the infection scene, we use live cell imaging to try and actually follow the interaction between these two in space and time to understand where and when things are happening during infection biology. Thank you.
Double click, Graham. Okay. Now, for aspergilloses, the diminishing usefulness of azole drugs is a serious health concern because the alternatives to azoles are few and they're flawed. So we're looking at some of Darren's work here, which has been funded by Gilead, and it illustrates the problem very nicely. So in all of these panels, we're looking at Aspergillus colonies, which have been genetically engineered to express a fluorescent protein. And these colonies are reacting to the application of drug in real time. On the left in green, we're looking at a fungal colony grown in the lab in the absence of drug. And in the middle and the right-hand panels in red and orange, we're looking at fungal colonies that have been treated with antifungal drugs that attack the cell wall, in the case of caspofungin, or the fungal plasma membrane in the case of ambisome. And so what you'll realise from looking at the caspofungin movie in red is that drugs that target the cell wall of the Aspergillus cells are able to burst the growing fungal cell tips, and you can see this in the movie by the disappearance of the fluorescent signal, but you'll also see that that fluorescence soon reappears, and what that tells us is that although the drug is able to burst the fungal cell is quickly able to recover and undergo significant further growth. So these cell wall active agents often don't kill the fungal cells. In contrast, in the case of ambisome, this is a drug that is rapidly able to kill the fungus, and you can see this because the fungal colony just becomes arrested and it stops growing, the fluorescence dies down. But unfortunately, these drugs are also toxic to the human host. And so what Darren will be doing in his next phase of work is to see how we can treat resistant Aspergillus species with combinations of existing antifungal drugs and novel agents to try to mitigate resistance phenotypes, but also dial down the toxicity to human patients. Next slide, please, Graham. Okay, so another way that we can get around the problem of antifungal resistance is by identifying other approaches to treating fungal diseases. Now, in bacteria, the problem of antibiotic resistance has already outpaced the development of new drugs, and this has led to a lot of interest in different approaches. And in contrast to drug discovery programs which have been conducted over the last half century, there's now a much heavier emphasis on research programs that focus upon vaccines or anti-virulence drugs. And in the autumn of last year, the Centre for Medical Mycology appointed a drug discovery technician, Rebecca Inman, to lead the drug discovery programs emerging from the science conducted at our centre. And I'll close the talk later on with some examples from my work about the way that anti-virulence approaches can work. But first, I just want to highlight that of course, the best antifungal defense on the planet is a fully functional, healthy immune system. And unlocking the secrets of those defenses, in particular, the balance between healthy tolerance of exposure to fungal spores and the active clearance of inhaled spores, is a highly active area of research at the centre. And we're now going to hear from 4 of the researchers who are working in this area. Thank you, Graham.
Hello, my name is Gordon Brown. I'm a professor in the MRC Centre for Medical Mycology, and I direct the centre. I'm an immunologist, which means that I'm studying how the human immune system is able to recognize and combat fungal pathogens. My particular interest is in how immune cells, like you can see in the picture behind me, are able to sense the presence of fungal pathogens, like Aspergillosis, when they infect the body. And what we've discovered some years ago, and I've been working on through my entire scientific career, is a group of molecules that are found on the surface of immune cells. It's called a receptor, that actually recognizes sugars in the fungal cell wall. And this recognition triggers protective immune responses. And my research is directed really at trying to understand the responses that this interaction triggers and how this response is able to trigger protective immune responses, with the ultimate hope that once we understand the mechanisms here, we could use these mechanisms to develop new ways of treating these diseases. Thank you and goodbye.
Double click on, Graham, to the next film, please. Thank you.
Hello, my name is Ivy Dambuza. I am a postdoctoral research fellow at the MRC Centre for Medical Mycology, University of Exeter. My research focuses on understanding how our immune system recognize and create a memory to microbes. We call this immune memory. Why is it important to understand immune memory? Because understanding the mechanisms involved in immune memory will help us in future designs of immune therapies, but most importantly, to vaccine development to fungi such as Aspergillosis. Thank you.
Double click on, Graham, thank you.
Hello, my name is Remy, and I'm a PhD student working under the supervision of Professor Gordon Brown at the MRC Centre for Medical Mycology. And the goal of my PhD is to characterize a receptor, which is a protein that allows your immune system to detect microbes. And these receptors, it appears to repress the immune response that your body generates when you are exposed to Aspergillus fumigatus. And although it might sound like a bad thing to limit your immune system, actually, because we are breathing in the spores of Aspergillus on a daily basis, you don't want your body to generate an immune response that is so strong that you would feel sick every day. And so I hope that my project can help us understand how the immune system balances mounting an inflammatory response that is strong enough to eliminate the pathogen and keeping it low enough so that you don't feel sick every day.
And again, Graham, double click, final video.
Hello, my name is Peter Cook and I'm based here at Exeter. And it's a great privilege of mine that I get to lead a research team down here. And we're really fascinated in understanding how cells in our lungs are so important at not only clearing fungi, but also understanding what goes wrong and when these cells actually start driving responses that we don't want. Things like asthma. And we think if we can understand this much better, this process, we could lead to better therapeutic strategies for the future. Thank you.
Next slide, double click. Great. Okay, so in the final part of the talk, I want to come back to this idea of anti-virulence approaches. This approach differs from conventional drug discovery programs because an anti-virulence approach, rather than killing a pathogen, seeks to disarm a pathogen. And this approach can place less selective pressure on a pathogen to mutate and become resistant to a drug. And so it's hoped that by identifying novel drugs which attempt to disarm pathogens, these agents can be used in combination with existing drugs to treat drug-resistant disease, but also can be used to mitigate the increasing incidence of antifungal drug resistance that we're seeing in clinic. So next slide, please, Graham. So with any antivirulence strategy, the first important question to ask is, well, what makes this fungus a successful pathogen of human beings? And we know that there are many dozens, if not hundreds of Aspergillus species that inhabit our planet, but more than 70% of all human infections are caused by one species, Aspergillus fumigatus. And we're still learning to understand what the important genes and proteins that are expressed by Aspergillus fumigatus are that are important for driving infection. And the whole genome gene deletion project that's ongoing in Manchester will be a critical tool to help us identify what those functions are. In my group, a long-standing research hypothesis has been that Aspergillus fumigatus is very good at quickly adapting to the different environmental conditions it experiences when it moves from the natural environment into the mammalian lung. And one signal in particular that the fungus is able to identify in host tissues compared to the natural environment is increased alkalinity. So the soil is quite an acidic environment compared to the lung, which is more alkaline. Next slide. So in this slide, I'm showing you the results of quite a simple genetic engineering experiment that was conducted by Margherita Bertuzzi a few years ago now when I was still in Manchester. And with this experiment, we simply rewired the genetic signalling pathway in Aspergillus fumigatus that allows the fungus to recognize the fact that it's growing in an alkaline environment. And we did this by making the signalling pathway sensitive to a drug called doxycycline, which we could add into the laboratory culture medium. And so on the left-hand side, you're looking at Aspergillus fumigatus colonies in the bottom row of the mutants, where the signalling pathway is now tunable according to the presence of drug in the media. And what you'll notice at alkaline pH is that the fungus stops growing as the concentration of doxycycline from 0 up to 10 micrograms per mil is increased. And this effect is mirrored in infected tissues from the mammalian lung on the right-hand panel. At day 6 post-infection of mammalian tissue with the tunable mutant, we were able to demonstrate that if we switched off pH signaling, we could reduce the number of infectious lesions in that lung by about 60%. And this gave us significant confidence that the pH signaling pathway will be a good target for novel antifungal drug discovery. Next slide. So in recent years, what we've been doing is to search for inhibitors of the pH signaling pathway. And with Bethany McCann over the last 3 years, BBSRC funding, working in partnership with industry, we found a number of novel chemicals which are able to do just that. And we're now working with industry and with funding from the research councils to develop a programme where we can synthesize bespoke inhibitors of the pH signaling pathway with a view to conducting preclinical studies to see how effective these new drugs might be. Next slide. Just a quick point to make about these novel agents is that if we combine them with existing antifungal agents such as caspofungin, that's the cell wall active agent I showed you earlier, In this sort of checkerboard data set, the blue colour indicates fungus not growing. So at high concentrations of caspofungin in combination with compound 1, one of our most potent new molecules, we are able to inhibit growth of the fungus. And on the right-hand side, in combination with itraconazole, if we attempt to treat an azole-resistant Aspergillus fumigatus isolate, the same scenario, our novel agents combine well with existing antifungals to limit growth of resistant strains. Next slide. Drug discovery programs are fraught with difficulties, and on the left-hand side there is a series of decreasing columns in a plot that shows the progression of novel agents from the candidate nomination through phase 1 and 2 clinical trials all the way through to approval as a new agent. And what you'll appreciate is that there's a very high failure rate of novel agents, and very often this is due to toxicity. There's a big road ahead of us in order to develop the new compounds that we've identified, but we're lucky enough to be in a space where we can work with industry specialists and contract research organisations. And the plan in the middle of this slide is the work plan for the next 3 years to take the novel agents through preclinical development and into a stage of advancement where we can begin clinical trials. For this purpose, being embedded in a centre which is interdisciplinary, and involves proximity to colleagues who are well versed in preclinical studies of drug efficacy and pharmacodynamics, and also the design and implementation of clinical trials is a real asset in progressing this work. And so a major arm of the centre's research activities over the next few years will be to identify more of these novel hits and try to move them through this valley of death and the preclinical divide. Final slide, please, Graham. I'd like to thank all of the researchers from the centre who pre-recorded material for the talk today, and Graham for effectively rescuing the slide deck, and to the audience for tolerating the technical hitches and for listening to the talk today. And here's a snapshot of all of my colleagues in Exeter captured before the lockdown. Just demonstrating an expanding centre of like-minded colleagues who all work on different fungal pathogens of humans. Thank you.